Good evening, everyone. Today's class is on exocrine glands and it is a continuation of the class on epithelium. Now you've all studied about the epithelium and you know that the function of epithelium are number 1, protection, number 2, filtration, number 3, absorption, and number 4, secretion. It is the secretory epithelium we are going to study in detail. The exocrine or the endocrine glands are the modifications of this secretory epithelium which developed during fetal life. The competency which we will be studying is identify the exocrine gland under the microscope and distinguish between serous, mucous and mixed acini. Before we go on to deal with this competency, let's first understand how the gland develops. What is the difference between the exocrine and the endocrine glands? What is the structure of the glands, and what are the various modes of secretion of a gland? During fetal life, the secretory epithelium, wherever it has to form the glands, tends to proliferate. So let's make the epithelium. This is the underlying basement membrane over which are present the secretory epithelial cells. Please bear with me. My eosin and hematoxylin are not exactly the correct colors, but when you make your diagrams, please use your eosin pencil and hematoxylin pencils. Now the epithelium, this is the epithelium. This is which epithelium? The secretory epithelium. This would be the underlying basement membrane, and this would be the underlying connective tissue. So let's make the fibers, and in between we would have the nuclei of the cells or the fibroblasts which are present, and the blood vessels would be there. Right? So you've had a class on epithelium, you have had a class on connective tissue, so you know how the structure looks like. Now this epithelium tends to proliferate. So it proliferates and starts invaginating in the underlying connective tissue. So this epithelium has proliferated at this particular area and it has invaginated into the underlying connective tissue and making the nuclei of only a small area. Soon this invaginates further in and canalization also takes place. By canalization I mean a lumen is also established in between this wall of cells which had proliferated. So it would be something like this. This is the epithelium. Now this would be the lumen which has developed, and let's put in the nuclei. Now some of these cells in the lower part modify to form the secretory part. That is the part which is going to secrete the secretions. The remaining cells modify to form a conduit or a duct from which the secretions would be released onto the surface, or it may enter another organ. You know that the pancreas drains itself. The pancreas drains in the second part of duodenum. So it may, or they may be something like the sweat glands. They secrete on the surface. So it may either the secretion may be either coming onto the surface or it may enter into another organ, and this secretion is entering by means of a duct. Such glands which secrete by means of a duct are known as exocrine glands. In contrast, in certain places, the epithelium loses its connection with the surface, and now it forms a cluster or cord of cells or a follicles of cells which get arranged together, and they get surrounded by capillaries. Now this kind of a gland has lost its connection with the surface. So there is no duct. It secretes its secretion in the underlying connective tissue, and by means of diffusion, it enters into these blood vessels, which are the fenestrated capillaries which are lying around it. This kind of a gland, which secretes its secretion directly into the blood, is known as an endocrine gland. So on the basis of the mode of secretion, the glands are classified as exocrine glands. These are glands which drain their secretion by means of duct, or an endocrine gland which releases its secretion directly into the blood vessels. These secretions are known as hormones, and they go and affect distant organs. The exocrine glands, again, release their secretory products, which could be proteins, they could be the enzymes, which are proteins, they could be lipids, for example, by the sebaceous glands, or they could be a mixture of both the things, release it onto an external or internal epithelial surface. That is, either they secrete their secretions on the outside to the surface or into another organ by means of a duct or a duct system. In contrast, the endocrine glands have no ducts. They secrete their secretory products, which we said are called hormones, directly into the bloodstream to influence the cells throughout the body. Now, there are certain other modes of secretion also. For example, look at this particular figure. Here, this cell is secreting its secretion, which is acting on the neighboring cell. Similarly, the cell is also secreting its secretion, which is acting on its neighboring cells. So this mode of secretion is known as paracrine secretion. In some organs, the cells release their secretions which act directly on the cell itself, and this is known as autocrine secretion. But in this particular lecture, we will be concentrating mainly on the exocrine gland. So on the basis of a structure, the exocrine gland may be made up of a single cell, that is unicellular gland, or it may have large number of cells forming a multicellular gland. Now, the simplest example of a unicellular gland is the goblet cell. And you've already seen the goblet cell in your epithelium class when we showed you the slide of trachea, which was lined by pseudostratified ciliated columnar epithelium with goblet cells. So you have rows and rows of columnar cells. Interspersed between them are the goblet cells. These goblet cells secrete mucus. If we were to draw a goblet cell, it is wider at the apex as compared to the base. So its shape is like the Harry Potter goblet of fire. So that is why, not that is why, but these cells are known as goblet cells. They have a nucleus which is present in the base, right? And surrounding the nucleus, in this perinuclear area surrounding the nucleus are present the organelles, the cellular organs. These are the rough endoplasmic reticulum, the Golgi apparatus, as well as mitochondria and other important organelles which are going to be present in a cell. Now, the rough endoplasmic reticulum forms the proteins which get glycosylated in the Golgi apparatus and get packed up into vesicles. These vesicles are then released and come and lie apically. These vesicles are known as mucinogen granules. These are known as mucinogen granules, and they secrete mucin. This mucin, when combines with water, forms mucus, which acts as a lubricating agent. So what is this mucus doing in the trachea? In the trachea, it traps the dust particles, It humidifies the air which is coming in, and thereafter the cilia which is present on the columnar cell sweeps the dust particles towards the nasal cavity. So it forms a the trachea has a mucociliary escalator, and the mucus for this mucociliary escalator is coming from the goblet cells which are lining the trachea. They are also present in the large intestines. You've seen a slide of large intestine also where we had columnar cells, and interspersed between the columnar cells with these goblet cells whose function was to again lubricate the passages. This is a high-power magnification of pseudostratified ciliated columnar epithelium of trachea with goblet cells, which has been stained with hematoxylin and eosin. These structures, which are marked G, are the unicellular glands, the goblet cells, which are seen interspersed between the columnar cells of the trachea, ciliated columnar cells of the trachea. Note here that the cell is broader at the apex, narrower at the base. The mucinogen granules which accumulate at the apex push the nucleus down towards the base. Now, here, however, the cells appear empty. We cannot see the mucinogen granules. This is because during multiple washing steps which takes place while we are staining with hematoxylin and eosin, the mucinogen granules get washed away. As such, the cells appear empty. In contrast, when special stains are used, which stain the carbohydrate moieties of the mucinogen granules, you can see the mucinogen granules being seen. This is the same slide in which the goblet cells have been stained with Alcian blue. So what you have to remember is that the mucinogen granules get washed away during hematoxylin and eosin staining because they are water soluble. Now, the simplest of the multicellular glands is the sheath gland. Now, in contrast to the previous slide, where we saw the goblet cells, which was the mucus secreting cell, interspersed between columnar cells, whose function was different from secretion. Here, in sheath glands, each and every cell is capable of secreting mucus. So, if you look at this, the lining of the stomach, each and every cell which is out here has the capability of secreting mucus. And the sheath gland is unique to the lining of the stomach. It is composed of surface epithelium in which every cell is a mucus secreting cell. All the remaining multicellular glands in our body have a more complex structure. We are talking about the exocrine gland. So, as we discussed earlier, there is a ductal portion, and then there is this secretory portion. So this would be the duct, and this would be the secretory portion. The secretory portion is further classified depending on its shape. It may be tubular. So we are using shape first for classification. Like the one in the example out here, may be tubular, or it may be what we call as Asana or alveolar glands. These are shaped like a flask. So the secretory portion is flask shaped or like a grape. Then there may be further classification. The tubule may be just a single tubule, or it may be branched. Similarly, the asana may be single, like in this particular drawing, or again, it may be branched. Now, depending on the duct, what is the branching pattern of the duct? It may be simple if it is unbranched, or compound when the ductal system is branched. So, how do we name the glands? Let's see in the next slide. Now let's start with the simple tubular glands, and the best example of that is seen in the intestinal glands of colon. Now look at this particular gland. It is also known as the crypt of Limburger. This entire portion is a single straight tubule which has no branching, And it's because the shape is tubular, so we call it tubular. And since there is no branching, then we call it simple tubular. Now, it is also said that the simple tubular glands, which we see in the intestine, the intestinal glands of colon, have no duct. This entire portion is the secretory portion. Now, one more type of the tubular glands is when the tubule part of the secretory portion becomes coiled. So now we have something called the simple coiled tubular glands. Now look at this portion of this figure. This is a slide of thin skin. You can see the hair follicles which are present, and out here is the eccrine sweat gland. You can see how the ductal portion is a downgrowth of the epidermis, which is seen out here. Then this is the duct of the gland. Thereafter, you have the coiling taking place of the sweat gland proper. Now, when we cut, so it would be something like this. So this would be the epidermis. Then we would have the ductal portion, and then the tubular part, instead of being a straight tubule, has coiled. Like this. So this is what is known as coiled tubular glands. And the best example of the coiled tubular glands are the eccrine sweat glands, which are found in the slide of skin. Now, when we cut sections of, we see various profiles. So if I was to cut histological section of this particular gland, what we would see is multiple profiles which we have to identify in these profiles, that which is the ductal component and which is the secretory component. So now come to the slides, the histology slides. This is the downgrowth, which is the sweat gland. The secretory portion of it is lying in the dermis, which is present. This is the ductal portion, which is lying in the higher, which is lying just from the, which goes from the epidermis through the dermis and comes to the reticular part of the dermis, so in the lower dermis. Now, when we cut, we see profiles. These dark staining profiles, which you're seeing, which are again lined by stratified tuboidal cells, are those of the ductal portion. And these light staining profiles out here, again lined by stratified epithelium, are the secretory components of this particular gland. So the next category is the simple coiled tubular glands. The first one was unbranched straight tubular glands. Now is the simple coiled tubular glands. Now the tubular component of the gland has got branched. So look at this. This is again seen in the stomach, in the fundus part of the stomach. The surface columnar epithelium cells dips below and forms these gastric pits. And opening in these gastric pits are these branched tubular glands. Now, simple because there is no branching seen at the ductal level. Then branched tubular, then branched tubular. That means the tubular, the secretory portion is branched, not the ductal system. Another thing which you need to remember is that these simple branched tubular glands, usually again we say that there are no ducts present. Another place where we see them are in the uterus. The uterine glands are also an example of the simple branched tubular glands. Now, instead of being tubular, the secretory part is acinar in shape, or shaped like a flask or a grape. So this is known as simple acinar glands. These are seen in the paraurethral glands. Again, now it's the acini which has branched. The duct is still single. So if we were to focus here, now look at this. Look at this. One acina, second acina, third acina. So the acini have branched. So branched acina entering via a single duct into the hair follicle. This is simple branched acinar glands. The best example of it is the sebaceous gland, again seen in a slide of skin, and also the mucus secreting glands of the stomach in the cardia region. Now let's talk about the compound glands. the compound branched tubular glands. This should tell you that not only is the ductal system branched, but so also the secretory portion. So how would this look like? So we would have something like, this is the surface epithelium which is dipped. This is the gland. This is the duct. Thereafter, there are multiple ducts, smaller ducts, which are joined together to form this single duct. This itself would be could be formed by other ducts, and then would be the secretory portion. So this kind of compound branched tubular glands are seen in the duodenum. So the Brunner's glands in the duodenum, which again secrete mucus, are the best example of compound branched tubular glands. See in this particular slide. Now this is at low magnification, so you're not being able to appreciate, but look at this. This is the ductal system. Thereafter, smaller ducts, and then the tubules would be seen. However, when we cut sections, you do not see tubules like the test tube shape which we've been talking about. Instead, what we see here is the spherical shapes which are characteristic feature of them. We have to identify whether it's a mucus secreting gland or a mucus secreting tubule or a serous secreting acinus on the basis of how the cells look like, which we will just see in a minute. So this is the compound branched tubular glands. The next category is compound acinar glands. Now again, that means the ductal system is branched, and then we have the acini which are present. Example of these are the parotid and the pancreas. So look at this. This would be the ductal system which is getting branched, and thereafter we can see the spherical profiles of the acini with the smaller ductules which would have joined together to form this duct and then the main duct. The pancreas and the parotid are the best example. In this particular slide, these would be the examples of the acini, and this is the ductal system of the pancreas which is getting started from here. And then these are the bigger ducts which are being seen. Now the last category is compound tubuloacinar gland, and the best example of it is the submandibular gland. Here, not only do we see the secretory part as tubules, but the secretory part is also seen as acinus. So the components which would be present in it are, first we would have tubules, the secretory part in tubules, the secretory part as acinus, and tubuloacinar portions may also be seen. So if this is the ductal system which has come in, we would have secretory parts which are like this, tubules. Then some of the secretory parts, if I was to take here, would be shaped like acinus. Again, the smaller ducts are joining together to form the larger duct. And some of these secretory parts could be shaped like this, tubule, and ending in an acinus. So there are three components which are present: tubules, acinus, and tubuloacinar. The secretory part can be of these three types, and the ductal system is branched. So this is compound tubuloacinar gland, and the best example of it is the submandibular gland. Now look at this. Now in this particular slide, let's start tracing. Now look at this. These are the acini which we are seeing, right? This out here would be the tubular part. Can you appreciate the tubule nature of it? And look at this. This is more spherical. This is acinus, and this would be the tubular parts of the particular gland. This would be the tubular part of the gland. And these are the acini, and then ultimately they are combining together to form these larger ducts which seem to be lined by the pseudo-stratified epithelium. But before we move on further, what I need you to understand, then in histological sections, when the particular organ or the viscera or the gland is sectioned, what we see. So this is the duct, right? And we said the duct is shaped like this would be the duct. and this would be the tubule, or this would be the acinus, or this would have been the tubular acinar secretory part. When we section them, when we cut them, what we see are the circular profiles. So this is a circular profile of this particular tubule, right? And now we call it as an acin, because when we see it like this, we will call it as a spherical structure, which we are going to be calling it as an acini. This is one example. Now this is the spherical profile, or this is the duct. You can see how the duct is going on. But when we take a histological section and we are seeing this portion on our slide, again we would see it as a spherical profile. So this would be the profile of ducts, which we are going to be seeing. And the acinus, this portion. Again, when we've taken a section, we look at it as a profile. When we see it in the profile, we will be calling it as the acinus. So whatever may be the secretion, if it is mucus secreting structure, secretory component, we are calling it as the mucus acinus. If it is secreting serous, we are calling it as the serous acinus. Now, how do we label it as a mucus acinus or a serous acinus is what we need to see. So whatever may be the exocrine gland, its secretion may be either serous or mucus. By serous secretion, we mean thin, watery, whey-like secretion which is rich in proteins. So that tells you that the cellular architecture or the machinery should be such that it can make proteins. On the other hand, mucus is viscous. As we've already discussed, it's a glycosylated protein which is secreted from the cell as mucin. The cell secretes mucin which gets hydrated to form mucus, right? So example of serous secreting acini will be found in the pancreas, which secretes the various enzymes, the parotid. And the mucus secreting would be found in the goblet cells. We've already seen mucus secreting cells as goblet cells, number one, and we would see profiles of the mucus acini in the submandibular gland. So how do these profiles of the cells, serous acinus and the mucus acinus, differ from each other? Because in the slide, we need to be able to identify them. So let's start with the serous acini. The acinus is nothing but the blind sac of the secretory unit, which we are now seeing as this spherical structure. The serous acinus is lined by pyramidal shaped cells. Right? These are the pyramidal shaped cells with a spherical nucleus which is present at the base. These cells around the nucleus, again in these cells, around the nucleus in these cells are present the rough endoplasmic reticulum, because we need to synthesize proteins. So rough endoplasmic reticulum and the Golgi apparatus. Now the ribosomes which are present in these rough endoplasmic reticulum attract, because of their phosphate groups of RNA, attract the basic dye. So in these particular cells around the nucleus, we see basophilia. We can't see the organelles, but the staining characteristic of these organelles tells us that what kind of organelles would be present out here. So the serous acinus shows basophilia. Now once the proteins are formed, they get packaged by the Golgi apparatus and vesicles are formed. These vesicles occupy, these secretory vesicles occupy the apical area. And they, because of their protein content, attract the eosin dye. And thus, the pyramidal shaped cells of the serous acinus shows apical eosinophilia. So there is a staining characteristic which helps us to identify what kind of structure is present underneath. So the serous acinus is a spherical acini which is lined by pyramidal shaped cells. The nucleus is spherical and present towards the base. It's present towards the base because apically it contains granules again. And what are these granules called? These are called zymogen granules, which are protein filled. Now, because of this, the staining characteristic of this particular cell is that it exhibits basal basophilia and apical eosinophilia. The lumen is small. It's a small lumen because of the large shape of the cells. In contrast, if we were to make a mucus cell, a mucus acinus, I'm sorry, it is lined by columnar cells. It is lined by a, sorry, oh, 1 minute. It is lined by columnar cells. Again, the apical portion will be filled with the granules, right? So the nucleus is pushed towards the base, is flattened and is present towards the base. These cells appear empty. They are empty looking cells because the mucinogen granules have been washed away during processing for H&E slides. As such, the lumen appears larger. So it has a larger lumen. If we were to compare a serous acinus with a mucus acinus, the mucus acinus is also comparatively larger because it is a section of the tubule, while this is a section of an acinus, so it appears smaller also in a slide. Apart from the serous acinus and the mucus acinus, they are also present within a slide, in some slides obviously, and within some tissues obviously, what is known as a mixed acinus. As the name suggests, this acini would contain profiles of mucus secreting cells as well as the serous acinus, serous secreting cells, serous secretion secreting cells. However, these serous cells are present like a cap or a half moon over this mucus acinus. And these, let me draw the nuclei first. These are the mucus secreting cells. And these are the serous cells, which are forming the serous demilume. And this is the mucus acini. Now, what is happening out here? What is seen is that these cells have both the serous and the mucus cells, which are lying next to each other. So, if I was to draw, ये होना चाहिए था. They would have been a serous cell. They would have been some mucus cell. They would have been some serous cell. And some mucus cells interspersed with each other. So let's give them the nuclei. ये ऐसे होता और ये ऐसे पड़े हुए थे. Now during fixation, when we do fixation, what happens is the mucus cells, look at this particular section. When we do fixation, the mucus cells expand and they pushed the serous cells away, right? And these serous cells form a serous demilume, which is sitting on this mucus acini. However, there are some intracellular canaliculi which are still connecting these serous cells to the lumen. Now, what is the function of this mixed acinus? The mucus secretion, as you understand, is viscous, right? And the serous secretion is watery. Now, to enable this secretion to flow through the duct, The mucus secretion is secreted, the serous secretion is secreted in the lumen, and the secretion tends to flow. So that is wherever we have thick mucus secretions being secreted, the cells also have what is known as the mucous acini have with them serous deminules. In reality, in life, the serous and mucous acini, the serous cells and the mucous cells are sitting next to each other. If we were to do some different method of fixation of the tissue, like rapid freezing, then we would get a picture like this. However, what we follow in our labs, this conventional fixation, which causes the mucous cells to expand and the serous cells to be pushed away. This results in forming what we call as an artifact, which is the serous demilunes. So you should know, when we see in a slide what kind of an acinus this is, what is its function, and why it is present. And you should also remember these small intercellular canaliculi, by means of which are running in between the two mucous cells. This is one mucous cell, this is running the other mucous cell, which is running in between the mucous cells, by means of which the serous cell secretes its secretion into the lumen. And why is it there? The role of the serous cell is to release the watery secretion for the viscous mucus to flow easily. Another structure which helps in the flow of the secretions are these myoepithelial cells. Now, sitting on top of the mucous acinus and also in the serous acini, but not on the ductal profiles which we will see these myoepithelial cells. These resemble smooth muscle cells and are present between the basement membrane of the acinus and the basal lamina of the columnar or the pyramidal cell. Are present these specialized cells called the myoepithelial cells, whose contractions which contract and help the secretions to move towards the duct, wherever the duct may be present. So their contraction will help the secretions to move towards the duct. So look at this. This is a myoepithelial cell which is present out here with the mix, which is present with the in the vicinity of the acinus. And where is it present? Between the basement membrane of the acinus and the basal lamina of the cell. Now, till now, we've seen that the secretions which have to be released by the cell are present in the form of vesicles within the apical portion of the cell. But how are these secretions ultimately released? Either to go into the duct or to go into the blood vessel in case of endocrine. So the mode of secretion would be, the mode of secretion of a cell would be either, on the basis of that, the exocrine glands would be classified as merocrine glands, apocrine glands, or the holocrine glands. Now, what do these terms mean? Now, look at this particular structure. Now, this is a typical acinus in which the vesicles have just got accumulated on the top. This vesicle thereafter fuses with the plasma membrane and releases its contents into the ductal system. This is the merocrine gland. They are the most common glands which are present. The exocrine part of the pancreas is the best example of merocrine glands. In contrast, look at this, the apocrine glands. These are, the example of this would be the mammary gland. Here the secretory vesicle comes, it pinches off with the plasma membrane into the duct. So the part of the cytoplasm is getting pinched off into the duct and then the secretion is being released. Whereas here the secretory vesicle was fusing with the plasma membrane and just the vesicle was releasing its contents into the duct. In contrast is the holocrine gland. Holo, that is, the secretory vesicles get accumulated within the gland and thereafter the gland undergoes programmed cell destruction or apoptosis to release its content. The example of this would be the sebaceous glands in the skin. So we have three modes of secretion of exocrine glands. These are the merocrine secretion, the apocrine mode of secretion, and the holocrine mode of secretion. And the glands would thus be the merocrine glands, apocrine glands, and the holocrine glands. Now, why do we need to study about glands and these mode of secretions and their structure? Now, you all must have experienced acne during your growing ages, or you may be still experiencing it, or you may have had a sty on your eyelid, right? So, all these are the clinical situations which you will be encountering when you enter into practice. Now, what is acne? During puberty, when the testosterone levels increase in the body in both the sexes, what happens is that there is increased production of sebum by the sebaceous glands as well as keratin. Now this blocks, because of this increased production, the ductal part of this particular sebaceous gland gets blocked. And as a result, the normal skin bacteria, propionobacterium, proliferates within this secretion and results in a localized inflammation which manifests as acne. So you will be seeing day in and day out normal in your clinical practice all these situations, and you need to understand what is the mode of secretion, what kind of a gland we are talking about, where is its duct, what happens to the duct, so that you are able to solve those, understand the pathogenesis which is taking place behind various diseases. Now we've seen the components of a gland. We know that it has a secretory unit and it has a ductal system. Till now, we were focusing on glands which were a part of a viscera. But let's talk about the larger glands which are present in the body. What are the larger glands? The liver, the pancreas, or the salivary glands. These are a few examples. Now, we are going to focus on the simplest of them, which is the salivary gland. And within that, we are going to understand the structure of the submandibular gland. Thereafter, we will go to more complicated glands. But first, let's understand what is the architecture of an exocrine gland or a gland. Each gland in the body contains two components. Number one is parenchyma, which is the secretory units, plus the ducts or the ductal system. And it contains stroma, which is the connective tissue which is present. Plus, it also contains the blood vessels, which is carrying the oxygen, the nutrients, and the waste products to and fro from the secretory units and the ducts, plus the nerves, which are usually the parasympathetic or the sympathetic nerves, which are going to bring about the stimulation of the gland, depending on what is its function. Now, if we were to take, say, the submandibular gland, this is the submandibular gland, and we pass a knife or a wedge through it and cut a section of it, what would it look like? So we would have something like this, the planar surface which we've cut, and this is the rest of the gland which is present. Now each of these glands is covered by a capsule. This capsule is the dense fibrous connective tissue which is keeping the gland together. From this capsule runs towards the interior septa. So the blood vessels and the nerves are being carried towards the interior by means of these septa. These divide the entire gland into smaller components called lobes. From the septa, further, we have smaller trabeculae or septas entering within the lobe. So this divides it into further smaller components called lobules. And within the lobules, out here would be present the secretory units and then the ducts which are starting from it. So what would be the structure of this? Let's study that. So from the secretory unit, say this is an acinus, would start the first duct, right? So if this is the acinus, it would be draining its secretions into the lumen of this particular duct. And this duct is known as the intercalated duct, which is lying within the lobule. From this, now the intercalated duct of this particular acinus and this particular acinus would join together to form a slightly larger duct, or it would continue into what is known as a striated duct. So the next division would be a striated duct. Now both this intercalated duct and the striated duct are present within the lobule. So they are intralobular ducts. These striated ducts of other acini, groups of acini, would join together to form another ductal system which would be running on these lobules, which would be running on these smaller, which would be the interlobular duct, जो यहाँ run कर रहा होगा. So the interlobular duct, which would again combine with the interlobular duct of this and some other septas and ultimately form the lobar ducts. And these lobar ducts would join together to form the main duct, which would be running. So they would join together to form the main duct. So the acinus, from the acinus, which is present out here, starts the intercalated duct and it leads into a striated duct, or the intercalated ducts would join together to form the striated duct. This intercalated duct is usually lined by simple squamous or low cuboidal epithelium. From them comes the striated duct. Now this striated duct has a special architecture. If we were to look at it, it would be lined with columnar cells, right, with its spherical nucleus, which is more towards the apex rather than the base. So that's a distinguishing feature. And present towards the base are these striations. Sorry, they should have been drawn with again, let me make a profile. This would be the profile of a duct, which is lined by columnar cells. So the basal portion, the basal plasma membrane, forms infoldings which appear like striations right here. Okay? Along this are present mitochondria. So this is involved in ion transport. And an more apically placed nuclei. So this would how a striated duct would appear. Right? This is the structure of a striated duct, which shows basal infoldings, and along these basal infoldings are present the mitochondria because it is involved in ion transport. Now these striated ducts and the intercalated duct are present within this lobule. So they are known as intralobular ducts. The striated duct is lined by columnar epithelium. The intercalated would be flattened squamous or low cuboidal. Thereafter, these ducts would join and ultimately form the interlobular ducts. इस lobule और इस lobule के बीच में run करता हुआ interlobular duct. Here, the epithelium would now become more complex. So it would become pseudo-stratified epithelium. And from here, it would lead into this duct which is running in between the lobes. So it is known as the lobar duct. This would be lined by, say, stratified columnar or stratified cuboidal epithelium, which would ultimately lead into the main duct. This main duct, again, as we move from here towards the superficial area where it's opening. So, say, for example, in the salivary gland, the submandibular salivary gland, which will be opening into the buccal cavity. So, as we are moving from the interior of the gland towards the buccal cavity, its epithelium becomes more complicated. It moves from stratified cuboidal to stratified columnar, and ultimately it would be stratified squamous non-keratinized epithelium when we reach near towards the buccal opening. So what we are going to see in a histological structure are the profiles of these various ductal system, profiles of the secretory unit, which depending upon the function of the gland may be serous or mucus. Then we are going to see connective tissue stroma, which is lying there, and the blood vessels and the nerves which are present within the gland. So let's see how a slide of a submandibular gland would look like. So this is the slide of a submandibular gland. Now this is the ductal system. This would be the connective tissue stroma. You can see the blood vessels which are present out here within the stroma. So this would be the ductal system. And since this seems to be this could be within the lobule, and these would be, or it could be the intralobular duct, or it could be one of these ductal systems which are lying. So it is the interlobular excretory duct, and this means this is one lobule and this is one lobule. Now, submandibular salivary gland is a mixed, so it secretes mixed secretions. That is, it secretes both the serous as well as mucus secretion. So what can we see? We can see profiles of the mucus acini. How do we recognize them? One, they are empty looking cells. They are pale staining. The nuclei is situated at the base, right? So these are the profiles of the mucus acinus. Then you can see profiles of the serous acinus because it's secreting both secretions. One, the serous as well as mucus secretion. How do we see these? These are darkly staining. They would have a basal basophilia and an apical eosinophilia. The nuclei is spherical and present towards the base, right? A number of profiles of these serous acini. Now, leading from this mucus acini would be a small intercalated duct. Usually in these sections, we will not be able to appreciate the intercalated ducts because they are very small, and they usually reach, and they're very small, and they're very, so we are not usually able to appreciate them. And then comes the striated duct. Now, this is showing here, this particular acinus is showing it in continuity. But what we would see more often are these profiles. Now, can you differentiate this ductal profile from the serous acini? Look at this. In this, the nucleus is present towards the apex, and you can see these basal striations which are present, because of which this is known as the striated duct. The striated duct would lead into this interlobular duct, which is present within the lobules, and thereafter we would see, when we would see more complicated epithelium, it would lead into the lobar and maybe the main duct in certain sections. Also seen here are profiles of the adipose cells which you are seeing out here. Now focus on this particular acinus. This is a mucus acinus, on the top of which is present the serous demilunes. We've already talked about what it is, right? So within a slide of submandibular gland, you need to draw the serous acini. First you need to see and then draw them. The serous acini. You can see the tubules, the mucus tubules. We talked about that it has three components. You have the acinar component, you have the tubular component, and you have the tubular acinar component. So this is the acinar component, which is secreting protein secretions. This is the tubular component, which is secreting mucoid secretion, and also present are the tubular acinar acini, which are secreting the mixed secretions. Then the profiles of the duct, and this, see, this which is what is running would be the connective tissue septas, which are dividing the gland into smaller lobules. So this would be one lobule, and this would be the other lobule, and this would be the interlobular septum, right? You can see profiles of the blood vessels out here, and that's it. I think we've discuss whatever. So you need to make this particular diagram in your histology files. This picture is from Diffure, right? And you need to be able to make a mucus acinus, a serous acinus, discuss the differences between them. Now this is how your diagram in the file should look like. It should be made in a circle. You should show us the profiles of the serous acini. You should show us the profiles of the mucus acini. You should also show us the profiles of the striated ducts. Right, so all these acini as well as the ducts should be seen. So they are trying to show here the apical eosinophilia, right? So serous acini, the mucus acini, the striated duct, you should be able, and you should form the mucus acini with serous demilunes, the connective tissue septa. Please do not just make a normal color. Please put in fibers and cells within the stroma which you are seeing around this gland. So this is how this particular slide should be done, and this is how the labeling should be done on one particular side of the diagram. So the major components of the salivary glands are, number one, saliva, right? So saliva, what does it contain? It contains mucus. It contains proteins, which is the amylase proteins, so that the process of digestion of carbohydrates starts within the oral cavity. So it would be producing amylase. So that would be the watery secretion which is secreted, which would be in the watery secretion which is secreted by the serous acini. Also, the serous acini would secrete lysosomes, which could control the bacteria in the oral cavity, right? It would contain, which would destroy the microbes and help in maintaining the normal flora of the oral cavity. It would contain the mucus, which would help to lubricate and buffer the oral cavity contents. Then also bicarbonate is secreted within the salivary gland, and also present in the salivary gland are the IgA antibodies. Now, ये सब bicarbonate कहाँ से आया, IgA कहाँ से आया? Let's see that. So the pathway of secretion would be the serous acini secrete the amylase and the lysosomes. The mucous acini secrete the mucus. Both the secretions enter into their intercalated ducts, which are present with it, where bicarbonate is secreted and chloride is absorbed from the secretions. From here, the secretions enter into the striated duct, which secretes potassium and bicarbonate and absorbs the sodium. So what is happening is bicarbonate is being secreted by the intercalated duct and the striated ducts. That is, it's modifying, not secreting, it's modifying the secretions which are being secreted. The bicarbonate content of the saliva helps in buffering the food which is there within the mouth. From these two intralobular ducts which are present within the lobules, that is the intercalated duct and the striated duct, the secretion now moves into the interlobular ducts, then into the lobar duct, then into the main duct, which is the Wharton's duct. And from here, that is intercalated duct, to the main duct, the Wharton's duct, the lining epithelium of this particular ductal system becomes more and more complex. It changes from simple squamous to locuboidal to ultimately non-keratinized stratified squamous epithelium of the oral cavity, which is present just at the tip near the opening of the Wharton's duct in the oral cavity. Now, where would you be seeing the histology of the salivary gland in your practice? So what we study is this condition which is being shown out here is a pleomorphic adenoma, which is a tumor, a benign tumor of the salivary gland. So this is this position of the submandibular gland, and in this gentleman, the submandibular gland is showing a benign tumor, which is pleomorphic adenoma, which is seen in, which is approximately 65% of all the salivary gland tumors. So when it was excised by the surgeon, this is how it looked like. This was the normal salivary gland tissue, and this was the tumor tissue, which was sent to you in pathology to look at it. So this here is the normal salivary tissue which you're seeing. So you need to be able to identify the normal, and out here is the big tumor tissue which is being seen, so that you can tell, number one, the surgeon that yes, you've excised the entire tumor and I can see the normal tissue also, and I can see the tumor margins, number one. And you can also, you should know how the tumor looks like, and what are the what is the structure of the tumor, to be able to decide the treatment which needs to be offered to the patient. So with this, we come to the end of this lecture. We will now be going into, in the coming days, you will be seeing the histology slides, where we will like to show you the simple tubular, the branch tubular, all those glands which we discussed. And also we will be seeing the first tissue slide, which is that of the salivary gland. So thank you, and hope we'll be seeing the histology section soon.